Assalamu alaikum warahmatullahi wabarakatuh. Today, inshallah, we're going to start answering, starting from page 26 in the microorganisms. Okay, so in this question, page 26 in the workbook, uh, he's saying that the yeast is a microscopic single-celled fungus. It's a fungus made of only one cell. Okay, and like all the organisms, of course, the yeast respires. So the yeast respires how? Respiration, it's breaking down of food to release energy. So it's going to break down food, which is glucose, to release energy, and a byproduct which is also released is carbon dioxide. Okay, we want to detect if respiration happens or not. So we're going to test for the carbon dioxide. We're going to collect the carbon dioxide gas and pass it into a test tube containing lime water. The lime water is an indicator for carbon dioxide. The lime water is clear. If carbon dioxide passed into it, it turns to milky or turbid. Okay, so first, explain why the glucose is added to the yeast in the test tube. Of course, the glucose, it provides food for the yeast. So the glucose, it is the food for the yeast. It provides food for the yeast. That's why we add it. So the yeast feeds on the glucose and then use it in respiration. It breaks it down to get energy. Okay, why there is a rubber bung in the test tube here? Why do we put a rubber bung? To prevent the gases from escaping out side. I want the carbon dioxide gas to be collected and to move and enter the other test tube. So I have to put a rubber band so that the gases won't escape um, outside of the test tube into the air. Okay, what happened to the lime water and explain. Explain why this happened. First, what happened? The lime water went milky or turbid. So why? Because the carbon dioxide, which is produced from yeast respiration, passed into it and turned it turbid. So I have to say that this carbon dioxide, it comes as a result of the yeast respiration. Then think about how you could use this apparatus to test the idea that the yeast respires more quickly when it's warm than when it's cold. So what are you changing in this experiment, warm and cold? So you're changing the temperature. How could you change this variable? So simply by getting two test tubes and you put one in a cold place like the refrigerator and another one in a warm uh, place, and then see, so you're going to repeat this experiment. One time you're going to put it in the refrigerator and another time you're going to put it in a warm place. But to make it a reliable test, we're not going only to make it twice. We can do it more than one time for each temperature. So we can, for example, prepare two test tubes and put them in the refrigerator and other two and put them in a warm place to have a reliable experiment. So we can say that we're going to prepare two or more, and more is better, test tubes and put them into different places such as a warm room and a refrigerator. Okay, what variables would you keep the same in this experiment? So I should keep the same the concentration of the glucose, the size of the test tubes, the mass of the yeast, the type of the yeast, the volume of the lime water. All those factors have to be kept the same to have a fair test. Okay, how could you measure how quickly the yeast is respiring? I'm using here lime water, so I can measure the time taken for the lime water to go milky or to go turbid. The faster that it turns milky, this means that the yeast is respiring faster or more. Okay, the next question. Which of these food is made using microorganisms? It's the cheese. Okay, I want here the type of the microorganism to make yogurt, the type, so it's bacteria. I want one example of a type of microbe that is a decomposer. So it can be bacteria or a fungi. You cannot write in this case earthworms because earthworms are not a microorganism. And he wants the type, so you're going to write bacteria or fungi. Why decay is important, and it's out of two. The two reasons are that decays remove the dead bodies and the animal waste. Also, it returns useful nutrients back to the soil, so it recycles them, and this increases the soil fertility. Okay, now Karim is investigating conditions that affect yeast. He's adding different amounts of sugar to the bread dough containing yeast. So I want a suitable prediction. So here he wants the prediction. You can please circle the word prediction because I'm searching for a prediction, not a conclusion. And I'm adding more or different amounts of sugar. So the prediction can be adding more sugar will increase the volume of the bread mix. But you cannot choose the more sugar, the more the bread mix, because this is a conclusion and he is asking for a prediction. Okay, here he plotted his results in a table. So what could be the conclusion? The conclusion is that the more the sugar added, the more the increase in volume of the bread mix. So the more the sugar added, the more the increase in volume of the bread mix, or the more the volume of the bread increase. Okay, as you add more sugar, the rise will be more. Write an explanation for Kareem's results. So the explanation is that uh, when you add more sugar, the yeast will respire more, producing more carbon dioxide, and that's why the dough will become bigger and bigger, because the carbon dioxide is what causes the dough to rise. So when more carbon dioxide is produced, the dough will go bigger or become bigger and bigger. So here the 3 marks is as you add more sugar, the yeast respires more. This is 1 mark, producing more carbon dioxide, the 2nd mark. So the dough will become bigger and bigger. Next. What is meant by infectious disease? An infectious disease is a disease that is spread from one organism to another. Okay, what are the symptoms of food poisoning? It's mainly vomiting and diarrhea. We have to draw a line here. The chicken pox, it's a virus. The tuberculosis, it's a bacteria. The athlete's foot is also a virus. Sorry, a fungus. So chicken pox virus, tuberculosis bacteria, athlete's foot fungus. Give an example of a disease spread by animals. So it's like the rabies, which is spread by the dogs, or the malaria, which is spread by mosquitoes. The cholera is a disease. It's spread by the contaminated water, the water that contains feces or waste from a person infected with cholera. So the water will contain the bacteria. So I want 2 methods to prevent the spread of cholera. First, I have to treat the water or the sewage, and then I have to provide people with clean water or with vaccination. Both are correct. 2 symptoms of influenza can be fever, sore throat, coughing, sneezing. I want to describe 2 ways to prevent the spread of the disease to the people she lives with. So because she have influenza, she should cover her nose and mouth when coughing or sneezing. She also should wash her hands regularly. She shouldn't share cups or her utensils with other people. Suggest why a surgeon should wear a mask over his nose and mouth. Of course, to prevent the microorganisms from the surgeon's mouth or nose to infect the patient. So I'm protecting the patient. So I'm covering the the nose and the mouth of the surgeon so that no microbes will go from his nose and mouth to the patient and infect him. Okay, what conclusion can you draw? This is statistics of the number of cases in different years of the people infected with cholera. So the conclusion that you can get from this experiment is the last one, which is many people were vaccinated in 203 than in 1993. That's why we have less people infected with cholera. You cannot say that the number of cholera decreased because, for example, in 2001, it's more than 2000. So it didn't decrease regularly. Okay. So just an explanation. So maybe more people are being vaccinated against cholera. Maybe we have an access to safe water. Maybe there is an improvement in hygiene. So any of these answers are correct. Okay, now I'm going to get some milk and keep it at room temperature for 5 days in a sealed container. And I'm going to measure its pH for 5 days. As you see, the pH decreases. Why? Because the bacteria is changing the lactose, which is the sugar milk, into lactic acid. This decreases the pH. It feeds on the lactose and uses it in respiration, so breaks it down to get energy, and lactic acid is also produced with the energy, which decreases the pH. And then she predicted the number of the live bacteria. So the number of the bacteria increased and then decreased. So, can we suggest a reason why the number of the bacteria would start to decrease after day 3? So, maybe because the pH is too low. So, this acid is killing the bacteria, the lactic acid which is produced. So, the pH is too low, or the acid is killing the bacteria, or maybe the sugar is not enough. It's the bacteria is consuming the sugar, so the nutrients or the sugar are becoming too low, so they are not enough anymore. Okay, next question. I want an evidence that suggests that there is still some live bacteria on day 5, and this evidence you should get it from graph 1. So on day 5, why still there is live bacteria? Because the pH is still going down. So this means that the bacteria is still changing, it's still respiring and using and breaking down the lactose into lactic acid, and that's why the pH is still decreasing. So as long as the pH is still decreasing on day 5, this means that the bacteria is still alive. Okay, on graph 3, I'm going to put the milk in the refrigerator and see what is the effect of the pH. So here I'm going to draw a line over this line. It's going to go like this. This line can go like this, for example, to pH 5. So I can make the line go like this, a straight line, and the ending of the line, it can be, for example, um, here at pH 5 on day 5, just a straight line. But why did I make it slower? Why did the pH didn't decrease as before? Because it's in the refrigerator, so the bacteria reproduce very slowly and respire very slowly. Okay, they are quite inactive. Okay, so here I want to draw the number of the live bacteria over the 5 days. I have two options: either to draw with a curve, a smaller one below the first one, because the bacteria that are alive are smaller in number that in the refrigerator than that it was at the room temperature, or I can draw it at a slower rate. For example, you can get it like this and go up slowly until day 5. It's going to start to increase. How can I get this? For example, when it reached 5 at day 5, okay, so the pH, for example, here was 5 at day 5. So let's see, the pH was 5, where when the milk was at room temperature, it was about here when it was 2.5 days. So I will reach the same number of live bacteria 2.5 days. It was the top. So I will make it the top, but not after 2.5 days, after 5 days, because the same pH level was reached in the refrigerator, but after 5 days. So the number of live bacteria will be the same, but not after 2.5 days, after 5 days. So I will draw a curve that goes up slowly and reach the top after 5 days. This means that the number of live bacteria increases in the refrigerator, but at a slower rate. Okay, and I don't want after 5 days because he said only draw for the first 5 days. Okay, now the bacteria are microorganisms and they are used in food production. So I have here some colonies of a bacteria. I'm testing the best ways for the bacteria to reproduce. So the results were shown as follows: without oxygen and with oxygen. As you see, in in the absence of oxygen, no colonies of bacteria were formed on the petri dish, but with oxygen, there were colonies of bacteria, and as you increase the temperature, the colonies of the bacteria, the number of colonies increased too. So how do you know that these bacteria need oxygen? Because colonies were only found when there is oxygen. Or you can say that no colonies are found in the absence of oxygen. So this means that the bacteria need oxygen to reproduce. If there is no oxygen, the bacteria didn't form colonies, so they didn't reproduce in the absence of oxygen. Okay, the colonies formed are of different sizes. How you could measure the size? You can measure the diameter using a ruler. Okay, I want to draw the results. You're just going to plot the results. Uh, in 0, you're going to plot the results from the table. So in 0, you have 5 colonies. In 10, you're going to have 10 colonies. 20, you're going to have 20 colonies, and so on. You're going to plot them as x's. Okay, so put an X or a dot surrounded by a circle, and then you're going to join the dots, each two dots, using a ruler. If each two dots together, you're going to join them as a line using a ruler. Okay, the number of colonies at 60, you're going to predict the number of colonies at 60 without oxygen. It's 0 because the bacteria didn't reproduce in the absence of oxygen. Then what about 60? At 50 degrees, the number of bacteria, the number of colonies was 21. So here you can write any number below 21. So the range accepted is from 0 to 20. Any number from 0 to 20 is accepted. Okay. The last question. Sarah is investigating making bread. So she's mixing flour, water, sugar, yeast, and she made a bread dough. And then she took 50 cubic centimeter of the dough. She put it in the measuring cylinder, and she added the measuring cylinder in a water bath at 30 degrees Celsius. And then she measured the rise in the volume of the dough after 30 minutes. Okay, and she repeated this experiment with different temperatures of the water in the water bath. And as you see, as you increase the temperature of the water bath, the rise of the dough increased up to 60 degrees Celsius, and then uh, the bread dough rises, but not as 60. So the maximum rise was at 60 degrees Celsius. This means that the yeast needs a warm temperature, not cold and not hot. Why? Because in the cold temperature, the yeast is inactive, and so it didn't reproduce a lot. And when it's too hot, uh, the yeast is killed at very high temperature. Okay, so use the table. And what was the question that Sarah investigated? The question was the effect of the temperature on the rise in the volume of dough. So the temperature effect on the rise of the volume of the bread dough. So what is the effect of the temperature on the rise in the volume of the dough? Because you have the volume at the start, at the end, and at the end, so you are calculating the rise in volume. Okay. Then she used the same dough from the same mixture. What else did she do to make it a fair test? So she left it for 30 minutes. She left it for the same amount of time. She gave the dough the same amount of time to rise. She used the same starting volume of the dough at the start, and she used the same amount of water in the water bath. Okay. Why would using dough from a different mixture make the experiment unfair? Because different dough might rise differently, and different doughs would contain different amounts of ingredients like yeast or sugar or flour or water, so the test will not be fair. I have to use the same dough so that the rise will be the same and it will contain the same ingredients. She plotted the results. So would you please get a relationship from the result? So we can say that as the temperature increases, the volume of the dough increased. The volume of the dough after 30 minutes increased up to 60 degrees Celsius. Then it doesn't rise as much. It rises, but not as much as at 60 degrees. So it's out of two marks. The first mark, you're going to say that as the temperature increases, the volume of the dough increased up to 60 degrees. The second mark is it doesn't rise at as much at temperatures above 60. Okay, or you can also say that it has a maximum volume at 60 degree Celsius. So that was the maximum volume. Afterward, the volume started to decrease. Okay. Um, she made a prediction that the volume of the dough increased because of the yeast. So how to test her prediction? Simply, she can repeat the experiment without the yeast, and she's going to see that the dough will not rise. So she's going to make sure that the rise was because of the yeast, because without it the dough didn't rise. So she's going to repeat the experiment without the yeast, or she can use different amounts of yeast, and she's going to observe that by adding more yeast the dough rises more and more. So this means that what makes the dough rise is the yeast. Okay, thank you very much. See you next time, inshallah. Assalamu alaikum wa rahmatullah.